Okay, hi. Welcome to Physionic. If you're new here, my name's Nicholas Verhoeven. I'm a PhD candidate in molecular medicine and a cell biology researcher, and that's actually what credentials I'm likely going to be focused on for this particular long-form video, and it's going to be an extremely long-form video because I think I spent maybe 2 to 3 hours putting together diagrams and the data from a paper that we're going to be investigating. And this paper tries to at least start answering the question of how do saturated fats, specifically one saturated fat that I'll get into, affect our muscle cells, and more so specifically in our muscle cells, mitochondrial function. You've heard of mitochondria in the past, and I'll give you a breakdown on more on mitochondria, as well as on insulin sensitivity and insulin signaling within the muscle cells. So this is going to be pretty detailed. It's going to be pretty in-depth. I would not be at all surprised if this ended up running for an hour or more. This is the kind of content that I think the long-form podcast really benefits from. Not that it's really a podcast anymore. It's really just on video. But I think that having room to breathe and really to sink our teeth into the data, every piece of data, as opposed to me just pointing out certain highlights that tell a story, is incredibly helpful. So with that in mind, let's go ahead and jump into this. Like I said, I've got quite a few different diagrams and explanations for you, so hopefully you'll be able to follow along. And if you can't, if you're live, of course you can ask questions for later. But if you're watching this after the fact, then feel free to drop comments. I try to respond whenever I can, whenever I see them on the platform. Okay, so the question that we're examining is, how does saturated fat affect muscle cells as measured by mitochondrial function and insulin sensitivity? And this information comes from a paper that is one of several papers that I'm going to be covering actually on this topic as I'm slowly going to narrow down on an answer. And hopefully probably in a month's worth of content, we're going to get closer to an answer of how to necessarily consume our food related to saturated and unsaturated fats in relation to insulin sensitivity and potentially mitochondrial effect. So this comes from a paper that is certainly not infallible. It has a few issues, but nothing too major. And I'll point those out as we go through. But it's called Protection from Palmitate-Induced Mitochondrial DNA Damage Prevents from Mitochondrial Oxidative Stress, Mitochondrial Dysfunction, Apoptosis, and Impaired Insulin Signaling in Rat L6 Skeletal Muscle Cells. So this is in a established type of research. So we're going to be looking specifically at muscle cells, and although they come from rats, I've actually looked at research in humans, and there's a lot of parallels here. But certainly don't take my word for it. We're going to be looking at that data as well in the future. So, as an introduction, let's go ahead and jump into this. I like how I put my picture up and then I have to take it away immediately. Okay, so mitochondrial dysfunction is related. I'm not saying that it's the cause, but it's certainly related to diabetes, cancer, dementia, and many other diseases as well. Some of them have yet to be, we know the diseases, but we don't know the impact that mitochondrial dysfunction might have. And mitochondrial dysfunction is going to be, it could be defined in many different ways, but it's essentially the lack of normal function of mitochondria. So what do they do? Well, one of the big ones that most people talk about is obviously cell energy. Another one is signaling. We'll be going into a little bit more on cell signaling within the cells in this video. And it also has an impact on the health of the cell in terms of trying to maintain if a cell should stay alive or if it should succumb to damage, and then for us to replace cells that are too heavily damaged. So mitochondrial dysfunction, function has a lot of different roles, and I'm certainly just mentioning a few of them. Other ones are in heme synthesis, for example, which is a really vital one for the health of the cell as well. There's a lot of different ways that mitochondria are helpful, but we're going to look at specific measures of mitochondrial dysfunction. So to give you a little bit more background, although 99% of your DNA, so the DNA, the genes that are kept inside of all your cells, or most of your cells, actually not all of your cells have a nucleus or mitochondria, but the vast majority of your cells do. And those cells have 99% of their DNA, which is the building block to create genes for all the different proteins that make up your cells, that make up your body. Those are kept in the nucleus. Now, about 1%, maybe slightly less than 1% of your DNA, of those genes, are kept in the mitochondria. This is related to kind of the theory that mitochondria, a long time ago, that mitochondria were part of a bacterium, and our eukaryotic cells consumed them, but instead of phagocytosing them like you would in the immune system and end up destroying those mitochondria, at that time bacterium, they ended up having a symbiotic relationship, and that's where we get this idea that mitochondria are at one point independent of the cells, and now they are part of our cells, which is kind of a cool theory to think about. But anyway, the DNA, what's unique about the DNA, or one of the unique aspects of the DNA in a mitochondria, is that they're circular, much like you would find in bacteria. So you've got circular DNA, and you don't need to know all these different proteins and whatnot. This is, although I am going to talk a little bit more on the next slide about this as well, but these are different proteins that are found in the membranes of the mitochondria. So the mitochondria has two membranes, right? I've said this before in many other videos. Your outer membrane, your inner membrane, and inside that inner membrane are these different proteins. So different complexes, you've got 5 of them. And these actually allow, in general terms, to produce ATP, or cellular energy. So you've got a lot of electron movements that go through these different proteins, and then ultimately that leads to the production of ATP. I'm not going to go into more detail than that. There's a lot of complexity just related to these proteins, and I'd certainly be happy to go into them more. But for the time being, that's all you need to know, that these proteins are used to generate ATP. Now, these proteins, these complexes, the complexes of multiple proteins all stuck together, some of those proteins are encoded, the information for which is held in the mitochondrial DNA. So if you have damage to this mitochondrial DNA, then you potentially have damage to these proteins, because it's going to end up generating mutated or damaged proteins that then get incorporated in the production of these complexes, and then the complexes aren't going to quite work. Think of it like a factory building a car. Well, if they continuously keep putting a bad clutch in the car, then the car overall may look okay, but when it's actually trying to function, it's not going to function correctly. So similar process could happen here as well. So different components end up getting in the way of the electron transport chain, which is these complexes working. Okay, so this is a schematic that I quickly, this is the same image, but here I've added these red splotches to kind of denote that there's damage that's occurred to these complexes. And with that, you get an offshoot of what's known as reactive oxygen species, which are ROS here. And really, it's more coming from complex 4, because with complex 4, you have the use of oxygen in the production, the eventual production, excuse me, of ATP. But the big point here is that if you have a bunch of damaged complexes, then you could have the increase of reactive oxygen species generation. I know a lot of people talk about ROS, or oxidative stress. Those are one and the same. Increases in oxidative stress are not a good thing, but you also don't want to have no oxidative stress, as weird as that sounds. And the main reason is because ROS can be used as a signaling mechanism to tell cells to do certain things. So you don't want none of this, but you don't want too much of it either, because these molecules, what they do is they interact with the chemical structure of, let's say, these complexes, or it doesn't have to be these complexes, it can be other proteins that are floating around in the cell. And because of their unstable nature, think of it like a fidgeting molecule that needs something. It's like it needs its fix. You think of it like a drug addict almost, which it's just it needs its fix. It's like sugar high or something like that. And once it has it, it calms down, but it does damage to everything around it. So these are obviously, if they're overly abundant, it's certainly not a positive thing. Okay, so now getting into the study. So there's a few unique things about this study. The reason why I brought up several of the things that I did is because they're going to come back as we go deeper into figuring out what's going on here. So the study was done in plated cells. So these are isolated muscle cells. So the results that we're going to get aren't going to be coming from muscle tissue, which may have fibrotic tissue in it, tendon tissue, all that stuff. It's just going to be focused on specifically muscle cells. And what they did is they incubated these muscle cells. So these muscle cells are typically held in a liquid, a media. And then here in this media, they are fed with a series of different nutrients that end up giving these muscle cells life so they can continue to sustain themselves even though they're outside of a body. And saturated fat is what they end up adding, specifically palmitate. So they're going to add differing concentrations of palmitate, the researchers are, to this media that's feeding the cells. And then we're going to be able to observe what changes occur within the cells. And I'm going to walk you through all that. But they added a second element to this, which is actually pretty interesting because it tells us a little bit more on the mechanisms as well, in that they end up what's known as transfecting. This is something I've done in the lab before. And it's really cool. It's one of the main things that I find really fascinating about molecular and cell biology, in that you can infect cells with plasmids, or what are known as essentially DNA coming that we've manipulated and we've put into the cells so that those cells then express a particular gene. So in this case, we're talking about a glycosylase gene. And this glycosylase gene, what it does is it repairs DNA. So we're going to see what effect it has when they infect these cells, and the infection should typically not have any sort of other negative effects because it's only the addition of this gene, and thereby the cells should be okay with that quote-unquote infection. I'm using the word infection because technically it goes from the outside of the cell into the cell, and sometimes we use viruses to do that. But usually we use what's known as a plasmid, which is not a virus. So they either add it or they don't add it, and then the cells will end up actually producing, because they have more of this gene around, they'll start producing this human glycosylase, which is HOGG, so human glycosylase gene. And then this protein will then go to the DNA, which I was talking about earlier, the genes that I was talking about earlier, and we'll start repairing these genes and repair the DNA. Okay, so again, this is going to be pretty important. We're mostly interested in this effect right here with palmitate, but this will add some mechanistic data that's going to be really interesting. Okay, so the first piece of data is the researchers, something that they have to prove is that if they infect the cells, or if they end up using a plasmid, transfect the cells, then they need to show that the infection actually worked, that the transfection actually worked. So they need to show that, yes, this gene is actually found in the cells. The cells are now expressing this gene. Otherwise, we can't just take their word for it. We have to actually show that there's some level of proof. So in this situation, what they've done is this is the increasing concentrations of the infectious agent containing this gene. And here, all we're looking at is just if there's a dark smudge, then, which is protein levels, then we know that there is that particular gene is being expressed, creating that protein. So at a low transfection level, you don't see much, really anything. At a little bit higher, then you start to see some of this gene, and at the highest point that they end up deciding to use it, you start really seeing a lot of this gene. So in the conditions where they actually infect the cells with this gene, we are going to assume that yes, this gene is being expressed and it is having its effect that it's supposed to have. Now what's unique here is that this GFP is known as a control. So in this situation, they're doing everything the exact same, but they're leaving the gene out. So they're still using the same vesicle, they're still using the same vessel to infect the cells, but there's nothing inside of that vessel. So then they want to know, okay, well, this at least shows that no, you're not going to get any expression at the same amount. You're not going to get any expression of this enzyme unless you specifically construct the vessel or the vesicle with this gene. And finally, this is specifically in mitochondria because they have this what's known as a loading control. Or really, I guess you could call this a loading control, but in this situation, they're also showing that, yes, this is in mitochondria. And the reason why we know that is because this particular protein is only found in mitochondria, cytochrome C. So, and it's consistent, it's not huge variation between these. So we're seeing this is good. It's good to see. So yes, the gene does get expressed in mitochondria. Then they do another one for the nucleus and another one for the cytoplasm. And they find that, yes, again, this gene is expressed in all areas of the cell. So just keep that in mind as we go forward, because it's not going to play a huge role in terms of where things are, but in one of the pieces of data it will matter. So just keep in mind that this gene is successfully expressed in all of these different areas of the cell, the nucleus, the cytoplasm, and the mitochondria. All right, so the first real piece of data that we're actually interested in is to look at mitochondrial DNA damage. And I mentioned that mitochondrial DNA damage is one aspect that you can look at, but most of the DNA occurs in the nucleus. So I've got this muscle cell, we've got our mitochondria, we've got our nucleus, and then I've shown that there's a lot more DNA or genes that are kept in the nucleus. And then if we were to zoom in more, that DNA damage, quite literally, looks like a break in the strands of DNA that make up the genes. So this is what we're measuring here in this situation. So they're adding 2 different concentrations of palmitate. So they're adding half a millimole of palmitate or 1 millimole of palmitate. So you don't really need to know millimole. But just know that in one situation, it's a lower amount than the other. That's really it. And here we've got our GFP. Remember, anytime you see GFP, at least as far as I can remember in this study, GFP means that they're not adding the DNA repair enzyme. Okay, so the glycosylase, the DNA repair enzyme, is not present anytime you see GFP. So here they're measuring specifically mitochondrial DNA. And with the addition of palmitate, what they see is an increase in DNA break frequency. So the amount of times that DNA is breaking is more frequent with the addition of palmitate. And this also increases with 1 millimolar as well as lower concentration. Now, when they add this DNA enzyme, which is going to be abbreviated as MTSHOOG1, this is the DNA repair enzyme. Again, looking at mitochondrial DNA specifically, they find that there's no increase in DNA break. So there's a rescue here that occurs. So obviously, if palmitate may be doing some sort of DNA damage, but this enzyme is then coming in and fixing the DNA so that we don't actually see any differences, which is pretty cool to think about. You got to think these molecules are actually actively working right now as you're sitting or as you're listening or watching. These molecules are going, millions and billions of times over repairing your DNA because we constantly have DNA breaking that's occurring. So it's a really cool thought to have. And then the other 2 bars here are actually the same exact experiment, but looking at nuclear DNA. And here we see that palmitate's effects are specific to mitochondrial DNA, so it does not seem to affect nuclear DNA. So that's really interesting because it gives us some level of specificity here. Okay, so now the next aspect of mitochondrial dysfunction that we're going to be looking at is ATP level. So ATP, as I believe I briefly mentioned earlier, is the mitochondrial way of generating energy. So cell energy is ATP. There's also GTP, but the most common is ATP. So here I've got the schematic of a mitochondrion producing ATP. So this is cellular ATP levels, and I should caution here that this is not the actual just production of ATP. This is the total amount of ATP, so picomoles per 1,000 cells. So per 1,000 cells, how many picomoles are you getting? Which is a minuscule amount because we're getting really, really granular at this point. And again, with the same conditions, 0.5 or 1 millimolar of palmitate added. Here we see GFP without this DNA break enzyme. We see a decrease, or a much lower level, I should say, maybe much lower level of ATP levels. Now here, I'll tell you, this is something that they should have had another control for. So in this situation, they should have had a condition, probably in some of the other situations that I'll go over as well, where they didn't add any palmitate, because it's important to be able to compare the palmitate to nothing, to just the regular media. So what would be the standard amount of ATP that would be found in the cells? So this could be a standard amount right here. We wouldn't know, right? But what we do know is that when we add the DNA repair enzyme right here, that we see this increase in ATP level. So that's interesting, but I don't want to call it disingenuous. I'd be hard pressed to specifically say just off of this data that ATP levels drop because of palmitate, because we can't say that because we don't have anything to compare against. But most likely that's what's happening. But they should have had another control here. All right, so the next one is to look at cell viability. So this is a measure of cell death. And here, again, this is just plated cells, so it's not like if you take palmitate, all your cells are just suddenly going to combust. And I will go ahead and put a disclaimer here. The other research that I've read that I'll be presenting on in the coming weeks, there are some interesting nuances that I think might explain why we have certain effects with saturated fat that seem really drastic, and it turns out that they're not that drastic when we look at just kind of general living. I'll keep that as general as I can for the time being, because I'm trying to sow the seeds of curiosity. All right, so looking at cell viability, so the higher the bar, obviously, the more cells have survived. So palmitate addition, again, the same concentrations. And here we see how many cells have survived after, let's say, 24 hours. And here we see that, let's say, maybe 75% of the cells are still alive with palmitate addition. But that still means that we've had a decrease. And this is, what's nice here is that they did this as percent of untreated control. So they don't have to have a control next to it, because this is all normalized to the control. So the control being the media, the cell media without palmitate being added. That was not the case here. This is in an absolute sense picomoles per 1,000 cells. So they should have had a control in this situation. That's why there's a difference between these two. So here they did a good job in that there is a decrease, there is an actual decrease in palmitate with the palmitate addition, and then there's a rebound once you still have, and the white bar is the DNA repair enzyme, a rebound, but not a full rebound. You don't see a complete amelioration of this effect. So there's still some cell death that's occurring, and at higher concentrations, there's even more. So maybe you're getting 50% cell death with palmitate alone, and then once you rescue some of that DNA, then it goes up to maybe 65-70%. With the DNA repair enzyme. So there is some cell death that's occurring with the palmitate condition. All right, so now I remember this schematic that I mentioned before with this production of reactive oxygen species, which the oxidative stress. Here they're actually measuring the amount of oxidative stress that's occurring. And this is specific to mitochondrial reactive oxygen species production. And again, this is based off of percent of control, which is nice to see. And here we see that there is an increase. So we've got 100%, and we see an increase with the palmitate alone. And we don't really see an added increase with more palmitate, but ultimately we still see, over a 50% increase in oxidative stress with the addition of palmitate. And that is, of course, rescued when we add the DNA enzyme, DNA repair enzyme. Okay, so now to prime you a little bit here. What we're looking at here is the mechanisms that we're going to be investigating next. And these are going to be important for cell stress, so as kind of downstream of mitochondrial damage or mitochondrial dysfunction, but also going to be priming us to look at insulin signaling. So here we've got this mechanism that I've created, and this is based off of what they've said as well as a few other papers that I kind of skimmed over. And ultimately you have this mitochondrion. It produces reactive oxygen species, so oxidative stress, as we saw right here. And then this actually leads to, not directly, but indirectly, you see an activation of a protein known as J&K. J&K is part of what's known as the MAP kinase pathway, or one of the MAP kinase proteins. And ultimately what happens is if it's phosphorylated, if it's activated a certain way, so if it's phosphorylated, a phosphate molecule is added to this protein, then it is activated. Now what happens when it's activated? Well, there's a bunch of downstream proteins that get activated or manipulated, adapted some way by J&K, which I sometimes abbreviate as junk, just because it's just like missing just that one letter. And then ultimately that leads to an increase in what's known as apoptosis related proteins. So these are known as the BCL2 family of proteins. And like BAX and BAC is something that I've studied in my lab actually. And ultimately what these proteins do is they end up puncturing the mitochondrion. And when they puncture the mitochondrion, a lot of the inner sections of the mitochondria will leak out into inside the cell still, but outside of these compartments that are held in pretty close. They have to be held within a particular section of the cell. So, you've got this inner mitochondrial membrane, outer mitochondrial membrane. Well, one of the proteins that I mentioned at the very beginning was cytochrome C. The cytochrome C would be found in that section. So if it ends up leaking out, then that's a sign to the cell, okay, we've got a lot of mitochondria that are dying. So as a result, this ends up potentially could activate a protein known as caspase-3. And there's a lot of different mechanisms involved here. This is not just caspase-3. There's a lot of other mechanisms, but the ones that they focus on are these, so that's why I'm focusing on them. So it's not as simple as this, just to be clear. So caspase-3 is a protein that's known as a protease. So this is a kinase in that it adds a phosphate, and this is a protease in that it will cut or cleave other proteins. And what happens is that this protein itself gets cleaved. So this cleaved caspase-3 is actually an activated form of caspase-3. So caspase-3 is like a lion without a target. It's just feeling lazy. It's just kind of floating around. It doesn't really have any distinct goal in mind, target in mind. But once you shock the lion, once you cleave the caspase, then it becomes active. And suddenly from that point on, it tries to destroy other proteins within the cell. And the reason for that is because it's trying to lead to so much damage within the cell that the cell ends up succumbing and ends up dying, known as apoptosis or cell death. So again, reactive oxygen species production, J&K activation leads to mitochondrial damage, lethal mitochondrial damage, then the activation of caspase 3, and then cell death. All right, so here's what we're looking at. Here we're looking at, again, the protein levels. So again, just if you see smudges, they exist, and the darker they are, the thicker they are, typically that's seen as more of that protein being present. Now, normally, let me just quick add here, normally you wouldn't, I've seen this worse in the past, but normally you would not want to have these be too blown out. So you don't want them so, like this one right here is really thick. You typically want to take readings that are thinner, that are clear, so that you can be able, it's tough to actually explain without actually showing you how this process occurs. But just overall, you typically wouldn't want these images to be so blown out. This is still probably okay, but I've seen some where all of these get so blown out that they end up kind of merging into one another. And then it's really hard to tell which one is which, as well as when you're reading these, when you're actually developing these, it's hard for me to explain. When you're doing this experiment, it starts out as a really tiny smudge, and then it continues to build as you keep reading the experiment. This is really basic. You're gonna have to take a molecular biology course, but just look up Western blotting or electrophoresis, and you'll know that as it continues to blow out, as it continues to grow, so it will maybe go from this teeny tiny smudge to this smudge, and it'll continue to grow until eventually it gets to this point. And eventually it saturates, so it can't get any blacker, it can't get any darker. And that's a saturation point. You don't want it to go, you don't want to take readings beyond that, because at that point all the other proteins can catch up and everything's uniform. And that can be deceiving, that can be deceptive when a researcher is trying to read these accurately. So do I think that's necessarily happening here? No, I don't, but I have seen it in the past, and it's something to look out for. So, okay, so that side vent, let's just move on. All right, so here we've got, again, we're measuring J&K. This is the total J&K, and here we've got the phosphorylated version of J&K. Again, J&K is a stress enzyme that could potentially, one of the mechanisms, one of the reasons it exists is to push mitochondria into end up opening up and damaging the cell. So in the control condition, we've got our total J&K, and then we've got our phosphorylated one. The one that we're most interested in is the phosphorylated one, because that's the one that is actually activated, as I mentioned earlier. So in this is our condition known as the control condition. So this is in just the regular media. We see a little bit of phosphorylation of J&K, but nothing nearly to what we see in the palmitate condition. And this is, again, without the addition of the DNA repair enzyme. Now, in this situation, we have the DNA repair enzyme. It's the exact same going on again. And we see that maybe there's something right there, but that could be some non-specific binding. The point is that ultimately in this condition, if we compare this to this, we see, nothing. So this DNA repair enzyme would then ameliorate this effect of phosphorylation of J&K. And in case anybody's interested in molecular biology, it's a good thing that they added this. This is called a loading control. So you want to make sure the researchers are adding the same amount of total protein. So total protein including actin, myosin, J&K, AKT, IRS, like all the different proteins that make up cells. If you add more total protein to one condition than the other, well, that's unfair, because then you're going to get a bad reading or an unfair reading. In this situation, this is used as a loading control, a single protein that doesn't change much, so that then you can compare, okay, this is comparable to this is comparable to this, and this is comparable to this, and therefore then all right, well, this is all on the up and up, presumably. And therefore, the effect that you're seeing here, or maybe I should say, the effect you're seeing here is not because you've blown out this one, trying to load a whole lot of protein to make sure that this is blown out as well. That's my point. So this is good to see. All right, now looking at this caspase, which I mentioned earlier. So the caspase 3 in its inactive form and its full-length form, and then cleaved caspase. Again, we've got the same conditions, and with the control, you see that the cleaved caspase is non-existent. However, with palmitate, you see far less of the full length because it's being cut into the cleaved caspase, and you see a lot of cleaved caspase. And again, with the addition of this DNA repair enzyme, you see that there's an amelioration of, you get rid of this palmitate effect. And again, actin levels, they're not exactly the same, but they're pretty close. So it's not going to be so dramatic that you wouldn't see an effect here. So this is all good. So here, again, you're seeing that palmitate has this effect in increasing more of this cleaved caspase. So it's mounting evidence at this point that you're getting a lot of dysfunction and damage that's occurring within the cells. All right, so let's switch gears to insulin signaling and insulin sensitivity, and to give you a brief education on how insulin signaling works. This is again, if you couldn't tell, let me get rid of my image here. If you couldn't tell, this is again a highly simplified version of what truly happens, but let's go through it anyway. So you have insulin that's outside of the cell. So let's say your pancreas ends up releasing insulin, and then the insulin will then bind to this insulin receptor, which is known as a receptor tyrosine kinase. And you don't need to know that, but I'd like to keep my lingo going as well, keep good practice for myself. So this receptor tyrosine kinase, what ends up happening is that it what's known as dimerizes. So these two sections will combine together and kind of hug each other. Aw, isn't that romantic? And then from there, they'll go through a process known as autophosphorylation, where they'll phosphorylate, so they'll add phosphates to each other. And then from there, that then recruits these proteins that are floating around in the cell or they're nearby. And one of those proteins is known as IRS, so insulin receptor substrate. And IRS will then be phosphorylated. This activates, just to be clear, not every time you phosphorylate something does it mean that it activates, but in this situation we're talking that way because it is the case. So this then activates IRS. You have a bunch of different molecules that get activated between this and this, but I didn't want to add all those because it just adds complexity for no reason. And then you get to this AKT molecule, which then gets phosphorylated, activated, and then you eventually, there's AS160 and there's a lot of other proteins that get involved. And then you have these vesicles that are floating inside the cell, and they will then migrate to the cell surface and deposit these transporters known as GLUT4 transporters. So once these GLUT4 or GLUT transporters in general are in the cell membrane, then you have glucose or blood sugar that can enter the cell, removing blood sugar from the bloodstream. And all of this is due to insulin binding, the insulin receptor. So a lot of cascading events. So with that said, what does it look like? I'm going to keep this up here so that you can keep referring back to it as we look at each one of these proteins to see what happens. All right. So here we're looking at IRS1. So this is this protein right here. This is the total version, and this is the tyrosine phosphorylated version where they added a phosphate to the IRS. So this is activated IRS. This is total IRS, or unphosphorylated. And here, in this situation, what they've done is they've added insulin or taken away insulin. So in this situation, you can't really measure insulin signaling unless you actually add the insulin itself, right? So that's what they've done. So the minus is no insulin, and the plus is with the addition of insulin to the cells. All right, so the control with no insulin, of course, you wouldn't get any activation of IRS, because why would you? There's no insulin to activate it on the outside. So at this point, then, you can look at the control. With the addition of insulin, you get some activation of this IRS, and the total levels seem pretty high. which is perfectly fine. And then palmitate, on the other hand, when you compare these two total levels to these two total levels, you see that there's a significant reduction in the total levels of IRS with the addition of palmitate, even without insulin. Then with the addition of insulin, there's very just minute, maybe not even at all, any phosphorylation of IRS. Now with this DNA repair enzyme, here, we again see that in the control, we see an increase in the phosphorylation. And here, if we compare this one to this one, with palmitate addition, we see a phosphorylation event that's occurring. And we also see that IRS levels are increased. Now, based on previous information that I told you in this podcast, if you would like to, you can pause this video and go back to one of the other Westerns, one of the other protein quantification measures. There is a problem here. What is that problem? Pause the video and comment below. And then I'll tell you the answer. I'll give you 3, 2, 1. The problem is that there's no loading control. So, great example of what I was talking about earlier. Without a loading control, I can't know for certain if they didn't add maybe 3 times the total protein in these two conditions, and they added 3 times less protein in these two conditions. And that's the reason why we see such low levels of IRS and no phosphorylation. That could be the case. They should have had a loading control here. This is a no-no. You don't do this. So this is a problem. But again, if we were to move forward with the assumption, because they do other they do other measures as well, which is good, but this should have been done, and the reviewers should have insisted on this. All right, so now we're looking at AKT. So with the phosphorylated AKT, and this is at the serine site 473, but the point is that it's phosphorylated AKT. And here we've got total AKT. Oh, look at that. We have a loading control. Kind of weird that they don't have a loading control here. But they have a loading control here. So, all right. So let's, even if we were to just dismiss this information, this data, let's move on with this data, which actually looks quite nice. In that, again, same conditions, you see that there's a nice healthy phosphorylation of AKT, which would then lead to greater expression or greater GLUT4 translocation. And you see the total AKT is very consistent across the board. You see that actin is very consistent across the board, so the total protein is perfectly normal. And you see that there's a diminishment in the phosphorylation with the addition of palmitate. It's still there, but it's definitely decreased. And then with the addition of this DNA repair enzyme, you see a slight return of this phosphorylation of AKT. So clearly repairing DNA has widespread effects that are quite good in terms of preserving mitochondrial function, as well as insulin sensitivity and insulin signaling. All right, but this is all well and good. This is to look at the actual signaling itself and all that stuff. And there's another piece of data where they measure the GLUT4, which is fine, but it just confirms more of this. But really, it'd be great to actually measure the insulin sensitivity itself. So when they add insulin, how much of this glucose actually enters the cell? This is important. So they look at 2-deoxyglucose, which is just a substitute for glucose. It's a good way to measure. It's an easy way to measure glucose uptake by the cells. So this is glucose uptake with insulin or without insulin, with the control, so the regular media or the palmitate media, and with the DNA repair enzyme or without. And here we see that this is the one. So this is what everything is, all of these are compared against the black bars. So the hash bars are compared against the black bars. And we see that with the addition of insulin, we see a massive increase in glucose uptake. Perfectly normal. That's exactly what you would expect. Then with palmitate, you see a drastic reduction. So when you compare that to here, of course, there's a drastic reduction. And then here with the DNA repair enzyme, you see that you shouldn't see that much of a difference in general with the control, but with the palmitate, you see recovery of that glucose uptake. So this gives us some more information that palmitate actually not just affects the signaling, but predictably also affects the amount of blood sugar that actually ends up entering the cells. And finally, this is the last piece of data before we finish this up, is to look at the exact same experiment, but they are using a J&K inhibitor. So J&K is the stress enzyme that, again, leads to widespread effects, but one of the effects is that it affects mitochondria negatively. And in this situation, J&K can also have gene-regulating effects. It can also have effects, potentially, what they're testing here is it could have effects on insulin signaling and glucose uptake or blood sugar uptake. So here we've got our control, and here we've got insulin that's added. And again, we see the same effect is happening. We see an increase in glucose uptake. With palmitate, we see a reduction in glucose uptake. And here we've got our palmitate plus this J&K inhibitor. So it inhibits this enzyme, the stress enzyme. And we see that there's a rebound in the uptake of glucose. So if you compare these two. Now, this is all well and good. However, they also show the mitochondrial DNA damage that occurs when you inhibit J&K. This is horrible data. This is not good data for two different reasons. Here the researchers are trying to convince you that J&K inhibition leads to, if you see a darker smudge, that means there's more of it. So in this situation, I'm not entirely sure what they're trying to get across here. They do a poor job explaining this, but mitochondrial DNA damage, so I don't know if there's supposed to be just more DNA here. That's completely unclear to me. But here we see that the control condition has more of whatever it is. I'm assuming it's just DNA in general, or the assumption is to be DNA, but this seems like a protein quantification, so it's very confusing. So that's not well explained, and they should have done a better job here. The second thing is that if you were to compare this smudge to this smudge, so with the J&K inhibitor to no J&K inhibitor, I mean, I could just squint my eyes and just kind of hope that this goes up. And I mean, maybe even if I objectively looked at it, I could say that this slightly increased. The problem is they should have done, again, a quantification of this. So they should have actually looked at this and done what's known as densitometry, where they actually use and produce some bar graphs so we can actually see the difference instead of kind of relying on our eyes and our bias to kind of tease this out. And the other thing is no loading control. Again, so this is not good data. I would throw this out. So we're just relying on this, and we're not relying on this at all. All right, so in conclusion, palmitate, which is a saturated fat, it's the most common saturated fat, increases mitochondrial DNA damage, oxidative stress, and general mitochondrial dysfunction in isolated, and I should add that this is in rat cells, in rat muscle cells. It also reduces insulin signaling in the same muscle cells, and this is something that I'll be investigating in human cells as well. I've already looked at some of the studies, and there's a lot of parallels, I can say that much. These effects are largely reversed when mitochondrial DNA is repaired. Okay, so that's a lot, that's and what's going to be exciting, what I already have the content written out, but I'm going to be investigating another study by this research group, and I'm going to be investigating and then they look at unsaturated fats. So they compare saturated fats just like they did here, and they're also going to be comparing unsaturated fats, and that's really interesting data. And on the other hand, we're also going to have some more studies specifically in humans to see if any of this translates from a mechanistic standpoint. Does it also translate to humans? And I'm going to leave you in suspense about that. I'm also trying to investigate some studies on unsaturated fats and its impact, because there's a lot of conflicting data out there. But hopefully, if I can string together several studies in a row, we can kind of start to move in a direction. So we'll do that probably starting next week already. So with that, thanks for stopping by. Hopefully you weren't bored out of your pants. If you enjoy this kind of content, then I'm glad, because we need more people like you that are willing to sit down and actually go through the entire paper, go through the data. And of course, the paper's linked for you, so you can look at it for yourself and critique for yourself, as well as my notes will be linked as well, so you can see some of my added notes that I threw in there. Clearly, this paper is not perfect. It's not. It has some issues. But are they massive? Are they to the point where we throw out the entire paper? No. But certain things that we would have to say, hey, I can't make any judgments based off of this. I think the other paper that they have, I already read it, already took notes on it. It's better for sure. I think it has a few small issues, but I think it's definitely better. So we're all learning. Anyway, thanks for stopping by, and until next time.